Hey gang, and welcome to Unit 12, the intestinal protozoans. Be prepared to identify protozoans from cyst and troph images. Make sure you're referring to your laboratory diagrams and in your textbook. Okay, so here we go. All right, at first there's some terms that you're going to need to know, so we'll go ahead and go over those. Let's go ahead and talk about cyst. Cyst would be a non-motile, non-feeding infective stage of a parasite, and protozoans come in two forms. They come in a cyst form and a trophozoite form. The cyst form is considered kind of a rigid stage, whereas the trophozoite is a motile, feeding, non-infective stage of a parasite. So our protozoans go from cyst to troph. Okay, we have excistation, which is the hatching of a cyst with the liberation of a motile troph. So that's when a cyst becomes a troph. And then we have encystation, when a troph becomes a cyst. This is the process of the external wall formation in order to resist drying and adverse environmental conditions. So now that you know the protozoans have two phases, the cyst phase and the troph stage, we can look at the life cycle of the protozoan. In step one on the left of the slide, what we have is trophs in the intestine multiply by binary fission. And in step two, which is above step one on the left, we have the development of the precyst. Here, this is sometimes called the rounded troph. Food particles and waste are lost here. So the organelles are lost here and the cyst wall starts to be secreted. In step three of the life cycle, we have encystation that begins. This is an external cell wall that starts to be formed. And then in step four, we have the infective cyst that is passed in the feces. In step 5, the human ingests the cyst from fecal contamination of substance. So something like water or food has been seeded with the infectious cyst. Okay. Then the cyst passes to the small intestine, and there we have excistation, where we have the transformation from a cyst to a troph. And then there we've got the trophs that proliferate. At that time, the motile feeding critters start to proliferate in the life cycle, step number 7. Let's talk a little bit about the anatomy of the protozoan. We have a nucleus or nuclei, which controls the reproductive and other cell functions. It's like binary fission. It's asexual reproduction where one body separates into two. The DNA duplicates and each clone gets one copy of the DNA or the genetic material. So that is the nucleus. It's very similar to bacteria and how they undergo binary fission. We've got the cytoplasm. This is also known as two layers, as the ectoplasm and the endoplasm. The ectoplasm functions as the skin, limbs, and mouth, while the endoplasm contains the inclusions and ingested food. Ingested inclusions could be red cells or bacteria. The Karyosome, which is also known as an endosome, is a large chunk of chromatin. This is a mass of chromatin present in all cysts and trophs, classified as either small, large, lumpy, either centrally or eccentrically located inside of the nucleus. It is a dark staining body. Then we have peripheral chromatin, which is basically nuclear DNA. It's nuclear DNA found only in the Entamoeba genus. So anytime you see peripheral chromatin in the nucleus, we know it has to be an Entamoeba. This peripheral chromatin is classified as either fine, coarse, even, or irregular. And I'm going to show you some pictures of this coming up. Then we have chromatoid bodies. Chromatoid bodies are found only in Entamoeba cysts. They are cigar-shaped or having frayed ends. And chromatoid bodies are basically RNA containing bodies in female germination cells. So you might be asking yourself, how do we determine the genus and species of a protozoan? The genus is determined by the presence or absence of peripheral chromatin and chromatoid bodies. If you have peripheral chromatin and chromatoid bodies, it means that it has to be an Entamoeba genus. Okay, the peripheral chromatin is positive and it has chromatoid bodies, it has to be Entamoeba genus. The species is determined by other characteristics like the Karyosome, peripheral chromatin, chromatoid bodies, cytoplasmic inclusions, the number of nuclei seen, and the overall size and shape of the parasite. So we'll talk more about that in upcoming slides. All right, how do we determine if we have a cyst or a troph? A cyst has a uniform shape and distinct cell wall. They're often round or oval in shape, and they have a very distinct cell wall that is rigid. A cyst often has more than one nuclei. For example, an E. coli cyst has several nuclei, up to 16. A cyst may have chromatoid bodies, and a cyst, especially in the case of Iotamoeba, might have glycogen vacuoles. If you have a trophozoite, you will notice that the trophozoite has no consistent shape, and that is due to its motility. Remember, the trophozoite is the motile form of the parasite. Some motilities might look like they are sluggish, directional, progressive, or directionally progressive. And the trophozoites are the feeding stage of the protozoan as well, and so you may see ingested inclusions such as food, like red cells or bacteria. Usually Entamoeba histolytica ingest red cells, and a number of your protozoans ingest bacteria, including Entamoeba coli. The trophozoites only have one nuclei that's easy to see as well. Let's go ahead and take a look at the anatomy of a protozoan cyst. Remember, a cyst is infective, non-feeding, and non-motile. We've got a couple different structures that I want to point out. The first one being cytoplasm. Cytoplasm is on the inside of the protozoan cyst, and then we have the ectoderm and the endoderm. The ectoderm, also known as ectoplasm, functions as the skin. Okay, it also functions as the limbs in locomotion and the mouth as in feeding. The endoderm is the inner portion of the cytoplasm. This is also known as endoplasm. Here we have thinner fluid that contains the food, vacuoles, and glycogen. Glycogen is where we store carbohydrates. Okay, then you can see in the image we have a nucleus, peripheral chromatin inside of that nucleus, and in this example it is thick and irregular looking. And then we have the karyosome, which is the black dot inside of the nucleus, and here we can see that it's kind of eccentrically placed inside of the nucleus. And then finally we have chromatoid bodies. You can't see any chromatoid bodies here, but oftentimes in Entamoeba genus you will see them. Remember, chromatoid bodies are just sections of RNA. So here we've got several pictures of protozoan cysts that I wanted to show you. The top left is an Entamoeba coli. Usually this critter is fairly large at about 15 to 20 microns. On the upper right, we have a Giardia cyst. To me, it almost looks like a football, where it's kind of oval shaped. You can see at least 3 nucleus in that image, and then you can see the axostyle, which is the spine. And that to me looks like laces on a football. So the upper right image is Giardia cyst. The bottom left image is Endolimax nana. Endolimax nana has 4 nuclei, but you can only see 2 here. And you can see that the endoplasm is separating from the ectoplasm, and that's usually because it has been placed on formalin. The middle image on the bottom is an Iotamoeba buchlei. That part that is staining pink there is the glycogen vacuole. Iotamoebas always have a large glycogen vacuole that is seen easily in the cyst form. And then on the bottom right, this is Entamoeba histolytica, where you can see we have a one nucleus, and inside that nucleus we have fine peripheral chromatin and a karyosome that's placed right in the center of the nucleus. Okay, and then there's no chromatoid bodies in that image, but there could be in Entamoeba histolytica. So the previous 2 slides talked about protozoan cysts. Now I want to talk about protozoan trophozoites. The trophozoites are the non-infective feeding and motile stage of the protozoan. Here, if you take a look at the anatomy, we have a cell wall and cytoplasm. We have ingested inclusions. Ingested inclusions could include such things as bacteria or red cells, depending on the species. We have a nucleus, and usually with the trophs we have 1 nuclei. And then inside of the nucleus or surrounding the nucleus we have a nuclear membrane. And then on the nuclear membrane we have peripheral chromatin, which is basically genetic material. That peripheral chromatin inside of this protozoan is fine and evenly shaped. We have a karyosome, which is the black dot inside of the nucleus, which is just a big chunk of chromatin. And then we want to look at motility. In this image here, we can see that this troph is stretched out and directional. So you can see that it is motile. It's no longer round or oval shaped. So that is the anatomy of a protozoan trophozoite. Here's a number of trophs on the trichrome stain that I want you to see. The trichrome stain is really nice because we can observe these protozoans on oil. So the upper left, this is a picture of Entamoeba histolytica, and you can tell it's a histolytica because of the fine, even peripheral chromatin. The cytoplasm is kind of lacy looking or frothy looking. And then we have the motility where the organism is actually stretched out and directional. That is pretty typical of Entamoeba histolytica. In the upper right image, we have a Chilomastix. You can see that it's kind of stretched out and pointing at the posterior, and that the nucleus has gone to the anterior. The reason it's shaped like that is because of its torpedo-like motility. The image on the bottom left is an Endolimax nana troph, and you can tell that it's an E-nana troph because it has a very large karyosome. They call that the lump of coal karyosome. And you can see the nuclear membrane, but the nuclear membrane has no peripheral chromatin. In the middle on the bottom, we have Giardia trophs, and they have kind of a leaf-like shape. It has two eyes, but those eyes are actually sucking discs. And you can see the axoneme or the axostyle, which is the spine that holds the flagella that goes right down the middle of the protozoan. And then finally on the bottom right, we've got Entamoeba histolytica. Entamoeba coli on the bottom right. And you can see that cytoplasm is very dense. We can see that protozoan is not very motile. It's very bubbly looking and dark. And you can see that the peripheral chromatin is uneven and kind of chunky. So these are some examples of the protozoan trophs that I'd like you to know. Let's go ahead and talk about the three varieties of karyosomes that you're going to see. The first one I want to talk about is on the left of your image. This is a small and centrally placed karyosome. This would be an example of an Entamoeba histolytica. In the middle, we have a karyosome that is large like a lump of coal, and that would be Endolimax nana. And then on the right image, we have a medium to large karyosome that is eccentrically placed inside of the nucleus. It's sort of offset, and that is typical of your Entamoeba coli. Okay? Let's go ahead and take a look at peripheral chromatin. Remember that if our organism has peripheral chromatin, it has to be an Entamoeba genus. Okay? The peripheral chromatin is nuclear DNA that I said is found in the Entamoeba genus. If you take a look at the image on the left, you can see that the nuclear membrane has very fine and evenly placed peripheral chromatin. That would be like what you would see in Entamoeba histolytica. In the middle, we have an Entamoeba coli, and you can see that the peripheral chromatin is very chunky, coarse, and uneven. And then finally, in Endolimax nana, you can see the nuclear membrane, but there is no peripheral chromatin on that, so it's non-existent. There is no peripheral chromatin in Endolimax nana. It's really nice when you see chromatoid bodies because chromatoid bodies are only in Entamoeba genus. So if you see them, you know it has to be an Entamoeba. There's usually two types of chromatoid bodies. They have the round and cigar-shaped chromatoid body like you can see on the image on the upper right. And then we have frayed or sharp-ended chromatoid bodies, which is what you're going to see for Entamoeba coli. Okay, only about 25% of E. histolyticas have chromatoid bodies, and only about 40% of the E. coli cysts have chromatoid bodies. Okay, but Entamoeba histolytica has the cigar-shaped rounded end chromatoid body, and Entamoeba coli has the frayed end chromatoid bodies. One of the best ways you can determine the genus and the species of the protozoan cyst is by taking a look at the number of nuclei. Okay, some protozoans have one, but most of the time you're going to see more than one in the cyst. Usually in the case of Entamoeba histolytica, you see 4 or less. In the case of Entamoeba coli, you're usually going to see 8 to 16 depending on the maturity of the cyst. Remember, not all nuclei are going to be in the same plane, so anytime you're doing parasitology and you're looking for ova or parasites, you always want to have your hand on the fine focus. Now, whenever you're in a laboratory, you want to have a general idea of which protozoans are small, which ones are medium, and which ones are large. Now, you don't need to know these sizes per se, like you don't need to know the small protozoans are 5 to 10 microns, okay? Medium protozoans are 10 to 15, and large protozoans are greater than 15. But I do want you to have a good idea of which are the small protozoans, which ones are the medium protozoans, and which ones are the large protozoans, okay? The small ones are Endolimax nana, Entamoeba hartmanni, and Chilomastix mesnili. The medium-sized protozoans are Entamoeba histolytica, Iotamoeba buchlei, Giardia lamblia, and Dientamoeba species. The large protozoans are Entamoeba coli and Balantidium coli. Okay, so this is helpful to know when you're in a lab. If you come across a very small protozoan, it's probably one of those 3. A medium protozoan is probably one of those 4, and a large protozoan is probably one of the last 2 on the list there. In order to see motility of the protozoan, you have to have a raw stool. Once you put them on formalin, they're no longer motile. But if you ever examine a stool with saline that is not preserved, you're going to see a number of different types of locomotion. For Entamoeba histolytica, you're going to see that organism very stretched out. We call this a progressive, rapid, directional motility, and sometimes you'll see finger-like projections. But a lot of times, the protozoan troph is just very stretched out. Entamoeba hartmanni is motile but not progressive, so it's not terribly stretched out. Entamoeba coli is kind of sluggish and bumbly. It's very non-directional and kind of doesn't know where it's going. It has very blunt pseudopodia. You can tell that it's motile because it is irregular looking, but it doesn't seem like it knows which direction it wants to go. Then we have Endolimax nana. Again, it is sluggish and non-progressive. Iotamoeba buchlei is sluggish and non-progressive. Giardia lamblia and Chilomastix, these are both flagellates, so they're going to be very motile and active. If you're interested in identifying what you think is a cyst, make sure that you note a halo around certain genus. For example, if there's a halo around Entamoeba, it's a pretty good clue that you're dealing with an Entamoeba cyst. Also note the number of nuclei in all your planes. If you have more than 1 nuclei, you're probably dealing with the cyst. Note the karyosome size and location within the nucleus. That's going to help you speciate the protozoan cyst. Note the peripheral chromatin. Note not only its presence, but also the arrangement and if it's fine or coarse. And then the cytoplasm. If you have any inclusions in the cytoplasm of the cyst, you should note that. Some inclusions of the cytoplasm would be chromatoid bodies or glycogen vacuoles in Iotamoeba buchlei. Now, if you're ready to identify a troph, what you can do is note the size and shape of the organism. A lot of times, trophs are going to look a little larger because they are usually stretched out or motile looking. Okay. Note the size and placement of the karyosome. If it's offset and in the center, it's an Entamoeba coli. If it's small and in the center, it's an Entamoeba histolytica. Note the nature of the peripheral chromatin. We have chunky and uneven peripheral chromatin, which is seen in E. coli. And then we have fine and regular peripheral chromatin, which is seen in Entamoeba histolytica. Make sure that you're taking a look at the cytoplasm for inclusions like ingested RBCs and bacteria, because as you recall, the Trophozoite is the feeding form. Note if the cytoplasm is coarse, fine, or vacuolated. You do want to look at the cytoplasm closely, especially if you're trying to differentiate between E. coli and E. histolytica. E. coli has a bubbly and dense-looking cytoplasm, where Entamoeba histolytica has kind of a fine or granular or lacy cytoplasm. Let's go ahead and start by looking at the amoeba protozoans. These are all the protozoans that are motile by pseudopodia. The first amoeba protozoan that I want to talk about is a major pathogen, and this is Entamoeba histolytica. This guy can give you bloody diarrhea, okay? This is the 3rd most common cause of parasitic disease. The cyst and the troph are medium sized at about 10 to 20 microns. The cyst may have a halo around it, and the cyst usually has 1 to 4 nuclei, but the troph only has 1 nuclei. You're going to see a hydrophobic halo on direct exams and stained slides, so be looking for that. There's a small central karyosome in the center of the nucleus, and that peripheral chromatin is fine and even. The immature cysts may have cigar-shaped chromatoid bodies, and the trophs may ingest red cells. That's especially true with why we have bloody diarrhea with Entamoeba histolytica. They have directional motility and very fine granular lacy cytoplasm. I forgot to mention in the last slide that with Entamoeba histolytica, there's a couple of nicknames for this disease. One is called New Delhi belly because it was first identified in India. And it does give you bloody dysentery. But I want to go ahead and talk about Entamoeba dispar now. Entamoeba dispar is not one that we're going to deal with in lab, so you don't need to know it for laboratory purposes, but you will need to know it for lecture purposes. Entamoeba dispar is a non-pathogen. It's non-invasive and indistinguishable from E. histolytica. You're not going to be able to tell if you have an Entamoeba dispar or an histolytica unless you see ingested red cells. If you see ingested red cells, it has to be an Entamoeba histolytica. If you don't see ingested red cells, it could be Entamoeba histolytica or dispar, and we might want to just report it. Entamoeba histolytica / dispar. Entamoeba hartmanni is another one that we're not going to view in lab. It's just too hard for new people to recognize Entamoeba hartmanni because it looks like a small Entamoeba histolytica. Entamoeba hartmanni is one that you'll need to be responsible to know the information about for the lecture exam. Okay, but we're not going to be dealing with this guy in lab. Entamoeba hartmanni is a non-pathogen. It kind of looks like a miniature E. histolytica. Its motility is non-progressive, so that's how it differs from E. histolytica also, because E. histolytica has very directional progressive motility. For E. hartmanni, the trophs are not going to ingest red cells, and the cysts are very small at less than 10 microns. E. histolytica is usually greater than 10 microns. Now, the trophs of Entamoeba hartmanni can be a little larger at about 8 to 12 microns. Let's talk about Entamoeba coli. This is a commensal organism, so it is not a pathogen. However, if you see Entamoeba coli in an ova and parasite study, you should probably keep looking because it means that your patient may have been exposed to other protozoan pathogens. Okay? The cyst for E. coli is large at about 15 to 25 microns, and it usually has more than 4 nuclei. In fact, in the immature cyst, you're going to see 8 to 16. In the cyst, you're going to see large, irregular shaped chromatin and a large eccentric karyosome, so it's offset inside of the nucleus. The immature cyst may have splintered chromatoid bodies, and this is seen in about 40% of the Entamoeba coli cysts. The trophs are larger. They demonstrate sluggish motility and blunt pseudopodia. The cytoplasm is much more bubbly and coarse, and you may see ingested bacteria. Now, that's different than E. histolytica, where you're going to see ingested red cells, but Entamoeba coli, you're going to see ingested bacteria. Now, Entamoeba coli is commensal, so it's not going to evade the tissues like E. histolytica does, okay? This organism happens to reside in the colon. So here I've got a couple pictures of the different cysts that are similar to one another. We've got Entamoeba coli on the left and Entamoeba histolytica on the right, and you can see they're both round. They both have that hydrophobic halo around them. The differences are the karyosome and the peripheral chromatin and the number of nuclei. So if you take a look at Entamoeba coli on the left, you can see that there is at least 5 or 6 nuclei in that plane. The peripheral chromatin is chunky and uneven, and the karyosome is a little bit larger and offset inside of the nucleus. For Entamoeba histolytica, you only have the 1 nuclei, but you can have up to 4. Okay, and here you have very fine and evenly placed peripheral chromatin. The karyosome, you'll notice, is small and is placed right in the center of the nucleus. All right, so here's some pictures of trophs. This is Entamoeba histolytica compared to Entamoeba coli on the trichrome stain. The three upper images are E. histolytica, and you can tell they're E. histolytica because of their fine and even peripheral chromatin, and you can also check out the cytoplasm that looks granular or lacy, and they are very stretched out and have kind of a directional motility. The organisms on the bottom row are your Entamoeba coli trophs. You can see that their cytoplasm is very dense and frothy, and you can also see that the peripheral chromatin is chunky and uneven. You can also tell that they are not very directionally motile either. All right, let's go ahead and talk about Endolimax nana. This is a commensal organism as well. The cyst is small at about 6 to 9 microns. It's often oval shaped, but they can be round as well. It just depends on how they're oriented on the slide. They're going to have 4 nuclei, but I can tell you, you generally don't see them because of the way they're laying. If they're laying on their side, you may only see 2 nuclei, but sometimes you can see up to 4. They have no chromatin at all, and they have granular looking cytoplasm in the cyst. In the troph, they are small as well at 8 to 12 microns. They have sluggish motility. They're more rounded looking. They have no peripheral chromatin around that nuclear membrane. And they have a large, very distinct lump of coal type of karyosome. It's very, very easy to recognize. Now, we don't want to confuse the troph with the Iotamoeba trophs because Iotamoeba looks very similar to Endolimax nana. I would recommend, if you're not sure if you have an Endolimax troph or an Iotamoeba troph, that you try to find the cysts because the cysts of the two species are very, very different. So here's a couple pictures of the Endolimax nana cyst and troph, and this is on the trichrome stain. On the left, you can see the cyst, and because there is no nuclear membrane, you don't really see the outside of the nucleus. You only see the 3 dots in the image of the karyosomes, okay? Now, there's actually 4 dots. You just may not see all 4 dots because of the way that it's oriented. On the right, we have the troph. The troph is very distinct and easy to see. You can see the nuclear membrane, and you can see that large lump of coal like karyosome. Iotamoeba buchlei is an intestinal protozoan. There's only a single species, and it is commensal and a non-pathogen. Usually lives in the large intestine. The cyst is medium at about 10 to 25 microns. There's only 1 nuclei in the cyst, which you may not be able to see very well. There's no peripheral chromatin and a large karyosome. But the thing that really jumps out at most people is that in the cyst you have a large glycogen vacuole. In the troph, they are medium size, sluggish motility with 1 nuclei, no peripheral chromatin, and a large karyosome. Be cautious not to confuse it with E. nanotroph. You may see ingested bacteria in Iotamoeba buchlei as well. So the Iotamoeba buchlei cyst is very easy to recognize because it always has that glycogen vacuole. The first image on the left of the Iotamoeba cyst is one on the trichrome stain, while the second image, just to the right of the first one there, is an image of an iodine wet prep for Iotamoeba, and you can see that the glycogen vacuole is pink because of the iodine. The troph has a large nucleus, or a large karyosome, and it will often ingest bacteria. The troph is coarsely vacuolated and bacteria may be ingested. Okay, so you might see that as well. Dientamoeba fragilis has to be one of my favorite pathogens, okay? This is a pathogen, and it is very common in Nebraska and in the Midwest, and it resides in the colon. Okay, the method of transmission for this organism is unclear, and there is no cyst form, which is really interesting. The infective stage is actually the troph, which is different than all the other protozoans. It won't invade the tissues, but can be a pathogen. It ingests mucus and bacteria. It's medium sized at about 8 to 20 microns, so that size varies quite a bit. It is motile but non-progressive. It has a leaf-shaped pseudopodia when it undergoes motility. What I like about Dientamoeba is, to me, it looks like a little sad person. They're usually oval, and you can see the nuclei on both very ends of the organism. And inside of the nuclei, there are usually 4 little chunks of nuclear material. To me, it just kind of looks like a little sad face. All right, let's go ahead and talk about the flagellate protozoans at this point. Giardia lamblia is a pathogen. In fact, it's the most common intestinal protozoan in the U.S. This is also known as Giardia intestinalis. Okay, so that's the same thing as Giardia lamblia. There are 190 different protein coats on the organism, and we make antibodies to each one of these. And as it changes coats, we can still get sick from Giardia lamblia. In fact, there's about 280 million cases of Giardia infections every year in the world. Okay, the infective stage is the cyst, and it causes traveler's diarrhea or giardiasis, or another name for Giardia traveler's diarrhea is called beaver fever, and that's because it comes from humans and other animals like dogs, cats, and beavers. It lives in the small intestine in the duodenum, and when you have Giardia causing you an infection, you have very foul-smelling diarrhea and burping and steatorrhea. Steatorrhea is basically fat in the stool, and people get what's known as the purple burps, and that's just because they're very foul-smelling. Okay, Giardia lamblia has a falling leaf motility. And the troph has what's known as a monkey face, and that's because of the appearance of the sucking discs, and I'll show you that in a couple images. The cyst for Giardia lamblia is medium sized at about 10 to 15 microns. It's usually football or pear-shaped. Usually the cyst is more football-shaped or oval-shaped, and there are 4 nuclei, although you may not see all the nuclei in one plane. They have no peripheral chromatin, but you will see the axonemes and the fibrils present. The axoneme is also known as an axostyle, and that's basically the structure that holds, it's kind of the spine that holds the flagella. Okay. The axoneme is an internal anchor for the flagella, and the fibrils are a small fiber of cellular origin. The troph of Giardia lamblia is medium also at about 10 to 15 microns. Usually you're going to see 2 anterior nuclei, no chromatin, 2 sucking discs, and several flagella. Usually there's 4. And the troph is going to be pear or spoon shaped. Now, when you're viewing Giardia lamblia cysts, you may notice that the cyst is pulled away, or the endoderm is pulled away from the ectoderm, and that is usually because of the way it is affected by formalin. So let's take a look at some pictures. Here you can see the cyst of Giardia lamblia. It kind of looks like a football. You can see the axoneme, a couple nuclei, and the fibrils. And then the troph has that monkey face appearance where you see 2 sucking discs, and it's kind of pear-shaped, and you can see the anterior flagella coming off the axoneme. You will also notice that the troph is very faintly staining when you stain it with the trichrome stain. Chilomastix mesnili is a small protozoan at about 6 to 9 microns. The thing about Chilomastix mesnili, it's so small, the cyst is so small, and you view the cyst on 40X, you may not be able to see too much except for the fact that it is lemon shaped. It has a little nub on it. Okay, and I'll show you some pictures here momentarily. There's no peripheral chromatin in Chilomastix, and the cytosome is basically a rudimentary mouth, and we call that the shepherd's hook. Okay, and I'll show you a picture of that as well. The trophs are small to medium at about 10 to 15 microns. Usually they're stretched out due to that torpedo-like motility. They can be pear-shaped or cone-shaped. They'll have 1 nuclei and 4 flagella that are anterior, and it'll also have a cytostome, which is the rudimentary mouth, and then the troph could also have what's known as a spiral groove, and that has to do with its torpedo motility. As it moves through the tissues and this stool, as it moves through those samples, it actually turns very quickly around in circles like a torpedo. It forms that spiral groove, and I'll show you that in a couple images. So here on the left, we have the cyst form of Chilomastix. The cyst, you can see the cytostome or the shepherd's hook and the nuclei in the trichrome stain because the trichrome stain magnifies it 1,000 times. When you're looking at it on a 40x, that only magnifies it 400 times, and so it's very difficult to see the nucleus or the cytostome. About all you can see is that it has a little nub and that it's lemon-shaped. It's a little bit bigger than yeast at about 10 microns. Okay, the troph is very stretched out. You can see that it's kind of pointy at the posterior end, and that has to do with its torpedo-like motility. You can see that it has one nucleus with no peripheral chromatin, and that has shifted to the anterior portion of the protozoan. You can also see a little tiny bit of a flagella coming out of that anterior portion just on the upper left. But usually it has several flagella that comes out of the anterior portion of the protozoan. I want to talk about the genital protozoan known as Trichomonas vaginalis, and we've talked about this organism before. There are several species of Trichomonas, but those are intestinal pathogens, while this one is a genital pathogen. Trichomonas tenax and Trichomonas hominis are both non-genital pathogens, okay? This is a flagellate. There is no cyst form. So very much like Dientamoeba, there is no cyst form. So the infectious form is going to be the troph. The transmission of Trichomonas vaginalis is by coming in contact or direct contact with trophs. It has one large nuclei, an undulating membrane that is noticeable. Okay, and is usually diagnosed by observation of the motile parasites in a vaginal wet prep. Now, in a vaginal wet prep, Trichomonas vaginalis is going to look jerky or twitchy. Okay, typically these organisms cannot survive the normal acidic pH of the vagina, which is around 4.0, and so they don't stay alive very long. Here's a couple images of Trichomonas vaginalis. You have 4 anterior flagella, an undulating membrane, the axostyle, and the nucleus. All right, let's go ahead and talk about the cilia protozoans. These are protozoans that are motile by way of cilia, and there's really only one that will affect humans, and that would be Balantidium coli. So let's go ahead and proceed to the next slide. Balantidium coli is the only cilia pathogen to humans. Now, pigs can harbor this organism, and we can actually come in contact with this ciliate by eating pigs. Okay, it is the largest protozoan at about 50 by 100 microns, so it's huge compared to the other protozoans. It is invasive, and it can cause ulcers. It lives in the large intestine and is invasive. It feeds on the host cells and can produce large ulcers. Okay, and this is a pathogen. So let's take a look at some pictures in the next image. The cyst of Balantidium coli is quite large and round at 50 microns in diameter. It has a very distinct macronucleus that's very easy to see. The troph is large and more oval shaped at about 50 to 100 microns. It also has a macronucleus that's very visible to see, and the cytoplasm can be vacuolated. It has a cytostome present, which is a rudimentary mouth, and you can see the indentation in the bottom left of the organism below, and that's the cytostome. And then it also has long cilia that are apparent as well, but it's tough to see it in these images, but they are ciliated. All right, we're going to wrap up today with the amoebic pathogens of the central nervous system, and that would include Naegleria fowleri and Entamoeba species. These amoeba flagellates alternate between the amoebic phase and the flagellate phase, so let's talk about those. Naegleria fowleri is an amoeboid troph that exists in humans. In the last 40 years, there have been about 200 people that have gotten this, and only 2 have survived. It is usually diagnosed post-mortem on autopsy from an examination of the cerebrospinal fluid. What happens is Naegleria fowleri lives in lakes and ponds and then enters the nasal mucosa, so it's sort of associated with swimming in contaminated water. It causes meningoencephalitis and can be fatal within 6 days. There's a little video on the bottom, a clip from Monsters Inside Me. If you want to copy and paste that into your browser, you should be able to watch a little clip on Monsters Inside of Me about Naegleria fowleri. Entamoeba is a protozoan of the central nervous system that has cysts that are infective and transmits by direct contact or inhalation. This organism has been known to cause eye infections and can actually destroy the cornea and can lead to meningoencephalitis. This is associated with poor contact lens care or contaminated water. Basically, this bug can go from the eye to the brain and cause central nervous system depression and meningitis. There's a link at the bottom for Monsters Inside Me. If you want to copy and paste that into your browser, you can watch a little video about Entamoeba species. So here on this slide, I have divided up the pathogens from the commensal protozoans. The pathogens are E. histolytica, Giardia lamblia, Balantidium coli, Trichomonas vaginalis, Dientamoeba fragilis, Entamoeba, and Naegleria fowleri. You can see next to the organism I put the infective stage. Okay, the commensal protozoans are E. coli, E. dispar, E. hartmanni, Endolimax nana, Entamoeba buchlei, and Chilomastix mesnili. So there are 4 parts of an ovum parasite study: the gross exam, the direct exam, the concentration, and the perm, or the permanent stain. Let's go ahead and look real quick at some comments that I want to make about the trichrome stain. So there are actually 2 stains available for ovum parasite studies. One is called the iron hematoxylin stain, and the other one is called the trichrome stain. From my experience, I have seen mostly the trichrome stain in clinical laboratories. In the iron hematoxylin stain, the protozoans stain bluish or grayish. They usually stain a little bit more grayish. The structural details are clear, but there is no color contrast, okay? So it's difficult to see the karyosome from the background material or the peripheral chromatin from the background material. The reagents that we use for the iron hematoxylin stain would be ferric ammonium sulfate, hematoxylin, phosphotungstic acid, ethanol. In the trichrome stain, we don't use as many unusual ingredients, okay? The trichrome stain is basically a 10-step staining procedure where our protozoan cytoplasm is either light green, pink, or purple. The trichrome stain provides contrast of internal structures, but every object that you will stain does not have the same color every time. Okay, the cytoplasm could be gray or purple one time and could be greenish or reddish the next time. It only provides contrast between structures, but not every structure is the same color every single time. Okay, karyosomes usually do stain red, and most background debris usually stains green, but that's not always the case. The stain provides color contrast for different internal structures, and the reagents used are trichrome stain, acidified alcohol, and regular alcohol. We usually use 100% reagent alcohol, and then our acidified alcohol is a mixture of glacial acetic acid and alcohol. And then finally, the last reagent we use is a blend of iodine alcohol as well. Let's take a look at the trichrome procedure. So this is the last slide for today's lecture material, but I just wanted to go over the trichrome staining procedure, and we're going to do this in lab. Okay, but the trichrome staining procedure is the 4th step in an ovum parasite exam. Here we're going to take a thin film of sample that's on a slide. Usually our stool sample is in a PVA preservative, and we take a scoop out with the little scooper and we put it on a slide, and we just want to make sure that it's a thin prep. After that's dried, we can put it in an iodine alcohol solution for 10 minutes. Then we're going to place it in 70% ethanol for 5 minutes, and then in another 70% ethanol for another 5 minutes. Then we place it in the trichrome stain. After we place it in the trichrome stain, we use the acidified alcohol to actually decolorize the slide. We're going to rinse it in absolute reagent alcohol by using quick dips, and then finally we're going to rinse one more time in absolute alcohol for about 5 minutes. Then we're going to place it in a xylene substitute like Periclear for 5 minutes, and what that does is it gets rid of a lot of the debris. Okay. Immediately while the slide is still wet, we can add a cover slip and apply the cover slip on there permanently. Once the cover slip is on, we can view our trichrome stain on oil, and then we report out parasites. We report them out as few, moderate, or many, or none. If you have few parasites, usually that is less than or equal to 2 parasites per oil immersion field. Moderate would be 3 to 9, and many would be greater than 10 or equal to 10 parasites per oil immersion field. All right, well, that's been our lecture on Unit 12, where we talk about the intestinal protozoans. Make sure that you're able to identify protozoans from images of cysts and trophs, and then make sure you're able to address all the other objectives for this unit. If you have any questions, please give me a call. I hope you enjoyed it. Bye.